So good afternoon, everybody. If I can have the first slide. Yes, thank you. This session is this session is entitled Insects and Other Organisms, and I will be telling you about viruses. So we've seen this slide before, so we know there's an extraordinary diversity of insects, and with all sorts of shapes, they evolved in a long periods of time. And we are particularly interested in the laboratory in the study of the interaction of these insects with infectious agents. And having colonized all terrestrial biotopes, of course, insects are exposed to a lot of microbes, often the same as we, so parasites, viruses, bacteria, and fungi. And the reasons to study the defense of insects against infections is mainly threefold. So the first reason is that they produce anti-bacterial or antifungal molecules, and by studying them, we can maybe use them as therapeutic agents, or at least by studying their mode of action, we can have ideas about new strategies to counter microbial infections. Of course, we share common ancestors, and so we've heard this in the talks yesterday on the use of insect and Drosophila as a model. So a number of genes are conserved, and by identifying important genes in Drosophila, we can learn things about human genes. And the last reason is that a number of hematophagous insects are vectors for infectious diseases. And these insects are not passive carriers. They are themselves infected, and so they fight these infections with their immune system. So in the laboratory, we are mainly interested in viruses. And this is another extremely interesting thing about insects, is that they are host of a large number of viruses. And this is a study that was published a few years ago by the group of Yun Zhang at the Institute of Disease Control in Beijing, where they have sequenced 220 animal species belonging to 9 phyla, and they have identified 1,400 viruses. And what you can see on the slide here is that a lot of these viruses are found in the arthropods, and in particular in insects. You have a very large number of insects. And if we look at another study that is this time focused on a particular family of viruses, the negative strand RNA viruses, what you can see is that you have here, it was 70 arthropod species that were sequenced, and they identified 112 viruses. And you have these viruses here, and what you can see is that insects are actually a reservoir for an extraordinary diversity of viruses, because you see that the viruses are extremely diverse. And what is interesting is that these viruses are at the root of many of the viruses that infect other organisms, including humans. So you can see, for example, here the flu virus, and you can see that it shares a root with insect viruses. You have filovirus here, the same, or RNA viruses here as well. So it is very relevant to investigate the interaction of insects with these viruses. And now, of course, some of these viruses can be transmitted to mammalian hosts. And this is an old slide from an article that related the history of the identification of new viruses, and you can see that a number of these viruses are arthropod-borne viruses transmitted by insects. And as we have seen since 2013, this was pre-Zika, for example, there are certainly a number of other viruses to come, hence the importance to understand why mosquitoes transmit viruses and why there are these epidemics some years, and then the year after it's gone, and we don't really understand why. And so what happens is that we have the urban cycle here where people get infected, and these viruses sometimes may spill over from a sylvatic circle where they are transmitted to wild animals. And so one of the parameters, we've all heard about the importance of the microbiome in a number of biological functions. And one component of the microbiome is also the virome, the viruses. And a lot of insects actually harbor viruses, as we've seen in the previous slides. So hence the question, could viruses persistent viruses present in mosquitoes be involved in the rate of transmission of insects? And so with my colleague and friend, Joao Marx, we started to look into that a few years ago. And so briefly, to isolate viruses, so the traditional technique is to filtrate sample tissue, to grow viruses on tissue culture, to use electron microscopy. And each of these techniques are not very sensitive, prone to bias. You may contaminate the sample when you work it. And so there is a direct method now, which is just to use high-throughput sequencing to identify new sequences. And this is what our Chinese colleagues have done in the papers I mentioned in my introduction. And in this case, you isolate RNA, you make cDNA synthesis, and you do high-throughput sequences. And then you compare the sequences you have to databases to find if you have viral sequences. So now I just want to make one introduction to the immune system of insects against viral infections. And this is a slide that summarizes the finding that we have made in our lab in Strasbourg, but also a lot of other labs in Europe and the US and elsewhere. And so basically, there is a major response to viral infections is RNA interference in insects. And basically, whatever the virus, you can find that they can be targeted by RNA interference. And then there are other genes encoding either restriction factors or signaling pathways, receptors that trigger inducible responses. And so first I will tell you about, in connection to this virome analysis, about this first response, which is antiviral RNAi. So the principle here is that when you have, so here it's the case of a positive strand RNA virus that enters the cells, the genome of the virus is like a messenger RNA. It will be translated, and this will produce the viral polymerase, which is an RNA polymerase, RNA dependent. And this enzyme will use the plus strand as a template to make the anti-genome, the minus strand. And then, in turn, the minus strand will become the template for the polymerase to make the plus strand. And in this intermediate step of replication, you have double-stranded RNA that is generated. And this is recognized by the enzyme DISO2, which is an RNase-free enzyme. And so DISO2 will bind to the viral double-stranded RNA, and this RNase-free will cut it, and it will generate 21 nucleotide long small interfering RNAs, or siRNAs. And this duplex will then be loaded on another enzyme named AGO2 with the help of this RNA binding protein called R2D2. And in AGO2, one of the two strands will be cleaved and discarded, and the remaining strand will remain and act as a guide. So it will provide the information that will allow AGO2 to recognize the viral sequences by base pairing. So it's an extremely adaptive system. The recognition of foreign double-stranded RNA will generate a small RNA that will program the nuclease to degrade the viral RNAs. So, and to show you how robust this is, you have here the small RNAs that are generated in two insects. So this is a virus that is called vesicular stomatitis virus. It's an arbovirus transmitted to vertebrates, mainly cattle, by insects. And here, this is what we obtained in laboratory in the fly. This was work that was done a while ago in collaboration with Carla Salé at the Pasteur Institute. And basically, what you have on the first graph here on the top, it's the size of the small RNAs. So basically, we sequence the small RNAs, and you will see that we have a sharp peak precisely at 21, which is the size produced by DISO2. And the second graph here, you have the abscissa here is the viral genome. And in blue and red, you have the number of small RNAs that were sequenced. In blue, when they match the plus strand, and in red, when they match the minus strand. And so basically what you can see is that the whole genome is covered, and you have the same density on the plus strand and on the minus strand, which is exactly what you expect if these small RNAs were produced from double-stranded RNA. Last year we published with Joe, we repeated the same analysis, but this time in one of the vectors, the natural vectors of VSV, which is Lutzomyia longipalpis. And what you can see is that the picture is very similar with the sharp peak at 21, and then the coverage, the homogeneous coverage on the two strands of the genome. And so the connection of this to the virome analysis is that this leaves us with a choice. If we want to identify viruses by RNA sequencing, we have the choice between looking at traditional RNA sequencing, where you just take long RNAs and you sequence them, or we can make the choice to just sequence these small RNAs. In other words, what is produced by the immune system. And the advantage of doing this is that then we look really at viruses that are replicating and that were recognized as replicating and potentially dangerous by the immune system. And so with João Marques, we compared with this group in Belo Horizonte in Brazil, we compared for the same samples long RNA sequencing to small RNA sequencing. And for most RNAs, as we might expect, we get more information by long RNA sequencing than small RNA sequencing. But when it comes to viruses, the picture is different. And here you have the size of the viral contigs that we could assemble from this sequencing. And what you can see is that in these three samples, the contigs are much longer when we do short RNA sequencing compared to long RNA sequencing. And as a result, when we try to identify the virus, we have a much better results with the short RNA sequencing. So here we have three viruses that were identified in these samples. And here you have the coverage with the long RNA-seq, and you can see there are lots of holes. It's very incomplete. But with the short RNA-seq, we basically reconstitute the whole genome, showing the promise of this technique. And now I'll show you briefly one application of that. And this is, again, work conducted under the directorship of João. So where we investigated a small city in Minas Gerais, Caratinga, and where mosquitoes were collected. And so a total of more than 700 mosquitoes were collected, and 31 of them were sequenced. And so basically we identified viruses in these samples. So one of them is Phasicaroen-like virus, a Bunia virus that was identified some years ago in Southeast Asia, so it is present in Brazil as well. The other one had been discovered by João and his team a few years before in the neighborhood of Rio de Janeiro. And the third one was a new virus, mosquito Caratinga virus. Using this technique of small RNA-seq. Now, this city of Caratinga was not taken by, just by chance. It's in a region that is endemic for dengue. And so now we can ask the question whether the presence of this virus might have an effect on the transmission of dengue. And so far, the data that we have is that when you look at first at the blood of patients that are available. So you find dengue in a percentage of the patients, but we don't find the other viruses, indicating that they are present in the mosquitoes, but they are not transmitted or they do not replicate in humans, which fits with the fact that we cannot replicate them and grow them on the mammalian cell lines. And when you look at Aedes aegypti, so you can see first that the prevalence of these viruses is quite high, and by contrast, the prevalence of dengue is quite low. It's the expected, what people report, in a few % of the mosquitoes are infected by dengue. But what is interesting is that if we look at the association of dengue with HTV or PCLV, we see a correlation, and we see a higher prevalence of dengue in the mosquitoes that contain HTV. This is highly significant, or PCLV, even though in this case it's less significant. Suggesting, of course, this is only correlation, so now we have to prove this in the laboratory, but suggesting that the presence of these persistent viruses, is not neutral and can have an impact on the transmission of dengue and maybe other arboviruses to humans. And so in summary for this first part, The information that we can get about biodiversity through these high-throughput sequencing techniques can help us to understand the dynamics of vector competence. Possibly it can help to prevent the outbreaks of novel viruses, because maybe some of the viruses that we identify can become new threats infecting humans. And also, when you look at the prevalence of these two viruses, PCLV and HTV, this could be one day turned into great vectors for manipulation of mosquitoes. So I will now switch to the second part of the talk. So I've talked to you now mainly on viruses and how they may affect the immune system of the host. And I want now to say a few words about the immune defense of mosquitoes against, and of insects against viruses. And again, I go back to this same slide. So there is the RNA interference. And then there are a number of genes that are induced by infection. This is something that we observed and others observed. And a few years ago, we decided to investigate one gene that we called the diedel. It doesn't look like anything known. And the reason we were interested in this gene is that, as you can see here, what you have here is the expression of this gene in flies infected by different viruses that belong to different families. And you can see that this gene is induced by 4 of the 5 viruses that are tested here. And in particular, it's very strongly induced by Synbis and VSV here. So this is now Drosophila. And so this was one reason to be interested in this gene. And the other reason was that when we looked at its sequence, it didn't look like anything that was, it's clearly not conserved in humans. So for a lot of grant agencies, it's not something that would be dubbed interesting. But when we looked at the sequence, first we were able to crystallize it and get the structure in collaboration with Alain Roussel in Marseille. So it's a new fold, it's a secreted protein, circulating protein. And we looked in the database, so here's the Drosophila protein, we found that it was conserved in the genome of several viruses. And not only that, it was conserved in granuloviruses, so like baculovirus, in ascoviruses, and even an entomopox virus. So this means that this did not occur once. It's known that cellular genes can be hijacked by viruses. And here, this gene was hijacked, but not only once, at multiple times. And not only that, we also identified a related protein in the venoms of some parasitic wasps, which instillate venom together with immunosuppressive substances in the larvae in which they inject their eggs. So this told us that this gene, there was probably something interesting, induced by virus, hijacked by other viruses. And so we decided to study infection in Drosophila. And to cut a long story short, I'll just show you the phenotype of the mutant flies. So we generated null mutant flies for diedel. And what we found is that these flies are short-lived. And especially when we infect them by one virus, Synbis, they die rapidly compared to the wild type. Interestingly, the next thing we do in these cases is that we look at the viral titer, because usually we expect that the flies die because they cannot control the virus, and there's a lot of virus replicating. And there was not more virus replicating here than here. So the reason the flies die, it's not because they do not control the virus. And so then we looked at the transcriptome of these flies, we looked at the genes they expressed, and here we had a very striking picture. We had a very strong, I will call it here for sake of simplicity, inflammatory response. So we had a very strong induction of antimicrobial genes, as if these flies were infected by bacteria. And it's known that these antimicrobial peptides, many of them are cationic. At high doses, they can have toxic effects, and all the more so here because it's persistent. It's not a transient response as you would have in a normal antimicrobial response. But these flies, mutant for diedel, they make all the time a huge amount of antimicrobial peptides. And so we suspected that this was the reason why they died. And so it was easy to prove it because there are 2 signaling pathways that regulate expression of these antimicrobial peptides. And one of them, the signature of the genes that we have was really coming from one of the pathway, which is a pathway discovered by Jules Hoffman and Bruno Lemaitre when he was in the lab, that is called the immune deficiency pathway. And so we combined the mutation of diedel with IKK gamma, which is one gene of the pathway, but we had the same results with IMD. And what you can see is that when we combine the diedel mutation with a mutation of the IMD pathway, we have a spectacular recovery of the flies, confirming that indeed there's nothing wrong with diedel not being there. Diedel is just there to control the IMD pathway, and if you block the IMD pathway, the flies recover. So the bottom line is that this told us that diedel was a suppressor of the IMD pathway, and you can learn a lot of things about immunology by looking at viruses, because viruses, when a pathway is bothering them, very often they find a way to neutralize it, and in particular by acquiring some genes. And so, for example, it's known that a lot of viruses have suppressors of RNAi, others have suppressors of apoptosis, baculovirus in particular, inhibition of translation. And so now we describe that several insect viruses have suppressors of the IMD pathway. So the corollary of this is that the IMD pathway must do something to viruses. And Akira Goto, a scientist from the lab, looked into that in detail, and he identified a function, not for the whole IMD pathway, but for two important genes of the pathway, the kinase IKK beta and the NF-kappa B transcription factor relish. So you can see that when you silence these two genes, there is a strong increase of replication of this picorna-like virus here. And so, to cut a long story short, we identified one gene regulated by IKK beta, which is a molecule called STING, and that we could prove that STING acts upstream of IKK beta in this pathway. And this is very interesting because STING is a very important gene in innate immunity in mammals. STING means stimulator of interferon genes, and there are no interferons in insects, but yet STING is present, and we showed that it regulates an induced response. And so, if there are no interferons, what are the antiviral genes that are controlled by this STING pathway? And Akira and Kiyoshi Ogado, who was a postdoc, just looked, undertook a small functional screen for genes regulated by IKK beta and to find genes that are antiviral. And here you have a loss of function screen, and what you can see that we have two genes here at the end. When you silence them, there is an increase of our replication, just as when you silence the kinase IKK beta. And here it's the opposite. It's a gain of function screen, so they overexpress this gene alone, and they look if it does something to the virus. And you can see that these two genes also, they reduce the virus. So one of these two genes is the factor STING. And now I'll finish by mentioning the other genes. This was an unknown gene, and so we call it NASO, which in the, Japanese means enigma. And so what is NASO? This is a protein with a single transmembrane domain, and I call it a Drosophila innovation. What is the reason? This gene is present in all animals. In humans, it's called C19 of 12. It has no known function for the moment. And the innovation is that in Drosophila, the gene has been duplicated, and so there are two copies. And so one of them we call NASO, and the other one that is more closely related to the gene in the other animal, so it's probably the ancestral copy we call father of NASO. And what is interesting is that there are a number of species of Drosophila for which the genome has been sequenced, and so we can exactly tell when the duplication occurred. And you can see that, for example, the subgenus Drosophila doesn't have the NASO. They have the father of NASO, but they don't have NASO. Whereas the Sophophora subgenus have both NASO and the father of NASO. And what is interesting is that when we overexpress NASO, so here it's in stable cell lines, so we induce expression of NASO here, and here you have the effect on the virus. And you can see that in each of these cell lines, each time we induce NASO, we have a very strong effect on the virus. We nearly completely block the virus. But if we do the same experiment with the father of NASO, so here we induce the expression, and you can see that we have absolutely no effect. So it was a duplication and a newly acquired function. And so I'm I believe that this is something that the insect field can bring. It's a very fundamental question. How do you build an immune system? And with this gene, NASO, now we have a very good system to ask how a gene became an immune gene using these fantastic tools of all the Drosophila species for which the genome has been sequenced. And what is really interesting, so you have here in bold the ones that we have now in the laboratory, and you can see what is interesting. So they all have sting and the father of NASO, but then some of them have NASO, but not others. And what is really interesting is that here you have their sensitivity to Drosophila C virus, this picorna virus that this pathway targets. You can see that the sensitivity varies. And you can see that even though some of them are very closely related, but they differ in their sensitivity. And what's also very interesting is that they differ in their ecology. And so we can see how the environment maybe helped to shape the repertoire of immune gene in these functions. And so I will start by this last slide. So we have on one hand this tremendous variety of insect viruses with common roots to the virus that plague humans. And then we have on the other hand this fantastic variety of insects that is not exploited to the moment, and genes like NASO, which is in the 1–2% of genes that are specific to one insect, they can have, as I demonstrated, some very strong antiviral effect. And so basically the message is that there is a very important diversity of antiviral genes and maybe also probably antibacterial, antifungal genes in all these insects. And this could be a very interesting challenge for future study to pinpoint what these genes are and how they work. And with this, I'll stop, and I'll thank my co-workers who are listed here. And as I mentioned, this is a collaboration with the group of Joao, who now also has a team in Strasbourg. Thank you for your attention.
Thank you, Jean-Luc. So we have a couple of minutes for questions.
Hi, Jean-Luc. Very nice talk. I wonder if you already found the counterpart of NASO in mosquitoes.
So there is a counterpart of NASO in mosquito. There is no, but there's no sting, which is intriguing. And so this is one thing that we would like to know. So first, in the Drosophila, we will focus on the Drosophiles for that. So there are species which have sting but don't have NASO. So then the question is sting antiviral? My suspicion is that sting will be antiviral. And then we want to look at what other genes are regulated by sting and mediate the antiviral effect. But in the case of mosquitoes, it will be also interesting to see if NASO is regulated by infection or not. But this we don't know for the moment.
Please use your microphone if somebody has questions.
Yes, I have a question on the first part of the talk. On the HTV, it belongs to Lithoviridae. It does not replicate in mammalian cells, and it has been, well, it's not infective in human, but it's a group of plant viruses also. So I don't know, I'm not virologist, if they infect other hosts, the HTV or the litho, if there are other Lithoviridae that do not infect non-plant hosts. And my question relates to the ecology of mosquitoes that are linked to plants because they are nectar feeders, and eventually they can also feed on other plants, etc. So they are dealing with plants.
So this is a very good question. So what the slide was showing is the hits you have in the gene bank. So one big limitation when you do metagenomics like that is that in the end you have your sequences and you blast them against the database, and it comes back with homology with a polymerase. And so the fact that the polymerase comes from, or the envelope or the capsid protein comes from one virus does not mean that the virus is from this family. So this is the first element. The second element is that you're perfectly right, and this is a big limitation also metagenomics is that when you find a sequence, does it come from maybe some integrated sequence in the genome? Does it come from indeed the food that was taken? And so this is what's interesting with this small RNA sequencing is that we focus on the 21 nucleotide reads, which is produced by the immune system. So if we detect it, it means that the virus replicates. And so indeed, there's no doubt that HTV is a mosquito virus and is replicating and persistent in the mosquito populations, even when they are taken to the lab.
Thank you very much, Jean-Luc. And we have to move to the next talk. And thank you, Jean-Luc, again.